To genetically engineer plants, scientists take advantage of the ability of plants to reproduce asexually. This process is called vegetative reproduction. One type of vegetative reproduction is test tube cloning, the process of growing a plant from a single cell or fragment from an adult plant. Since an adult plant can be grown from a single cell, genetic engineering in plants is easier than it is in animals. Scientists take cells from an adult plant and manipulate the DNA. The cells then grow into. Scientists have several ways to manipulate the DNA in a plant cell. To add DNA to wheat, rice, and corn, a DNA gun can be used. A DNA gun fires tiny metal pellets coated with DNA fragments. When a pellet enters a cell, some of the DNA fragments enter the nucleus and are in. A strain of rice called golden rice was generated using a DNA gun. The rice was engineered to contain extra iron and beta-carotene, a precursor to vitamin A. Golden rice can help the 100 million children worldwide who are vitamin A deficient. It can also help the 400 million women of childbearing age who are iron deficient, which places their babies. In other plants, genes of interest are introduced into the cells using a TI plasmid. A TI plasmid contains a stretch of DNA called tDNA, which inserts into the plant DNA. Restriction enzymes are used to place a gene of interest in the middle of the tDNA. When a TI plasmid is introduced into the plant cell, the tDNA and the gene of interest are incorporated into the plant's genome. A TI plasmid was used to create caterpillar-resistant tomato plants. Scientists discovered a protein produced naturally by the bacterium Bacillus thuringiensis that kills caterpillars when ingested. The protein is harmless to humans. A TI plasmid was used to introduce the gene for the toxic protein into a tomato plant cell. The genetically engineered cell was grown into an adult plant that's resistant to caterpillars. Cells from the engineered plant are grown into more caterpillar-resistant tomato plants.